Welcome to session 3 on environmental management and AMR. My name is Bart Smets. I'm at Aarhus University in Denmark. And first I'd like to introduce my co-chair, Joanne Elmos-Nino from Pfizer in Connecticut, USA. We'll have a number of outstanding presentations, and it's my pleasure to introduce our first speaker, and that is Professor Caroline Duchaine, who is professor and full professor in the Department of Biochemistry, Microbiology, and Bioinformatics at the Université Laval, and has a Canada Research Chair, Tier 1 in bioaerosols. And her work focuses on the study of bioaerosols in a variety of contexts, human, animal, industrial health, as well as on the development of analytical strategies for both natural and in vitro environments. Her research is highly transdisciplinary and involves collaboration with various experts, including physicists, medical doctors, veterinarians, engineers, and bioinformaticians. And I look forward to hearing Caroline present her presentation on bioaerosols and antibiotic resistance gene transport, understanding the role, impact, and fate. Welcome, Caroline.
Thank you very much for the introduction, and thanks you all for being here today. I'm very humbled to be given this opportunity to talk to you about bioaerosols, because I'm not an AMR expert, so I would like to apologize to start with, because you may not learn a lot about AMR with me this morning. However, I hope that I will maybe show you how important the air is in the transport and dissemination of antibiotic resistance in the environment. So I'm very thankful for the organizing committee for this opportunity. So you all know way better than I do about AMR, but what about AMR in the air? So what do we know about that? So we know that AMR can be found in any environment in the air. It can be indoor and outdoors. And there's quite a lot of data regarding antibiotic-resistant genes concentrations in various types of environments. However, from what we observe, there is almost no data on emission rates from point sources. So we will have a look at this today. Where bioaerosols can be emitted outdoor from a point source and also spread over long distances. So it's very challenging to understand the exposure and also quantify and evaluate the exposure to humans, of humans to those contaminated bioaerosols. So when it enters the airborne route, it can, of course, reach the lungs, but it can also deposit on surfaces in the environment. And through the airborne route in the lungs, it can also enter the digestive tract, because most of the aerosols that we breathe in will be eventually swallowed. And finally, it's been hypothesized that there is a long-term exposure that may occur and can represent a significant contribution to the AMR exposure. So last year, two major organizations, the WHO as well as the Canadian group of experts, have published both a research agenda or a priority agenda for managing AMR risk in human and animals. What is very interesting is both very well prepared and very serious documents that manage to put the priorities, determine that the environmental surveillance is of primary importance. We realize that the surveillance of the air is, however, neglected, not because I think the community doesn't think it is a priority. I think it's certainly because there is very few people that have the expertise, but also have the equipment and the resources to do the air surveillance. And in the project I will be presenting this morning, we will probably give you some options if this is something you're interested in. So the first question we were asking ourselves is, how could air contribute to an eventual risk in comparison with the food and water and surface exposure? So there was a manuscript published a few years ago that proposed those estimations from the airborne route and compare it with the other routes of exposure. And when we include the inhalation rate, the exposure frequency, exposure time, etc., those authors come up with some dose estimation that are attributed using the data in the literature that is available and that give ARG concentration in the air. So what those calculations led to is comparison of eventual dose from the water, from the food, but also if you are exposed to the air of various types of environments. So this is just one example that what the type of interpretation that can be done using air data of ARGs. But there's quite a few things missing in the estimation of the eventual exposure or eventual risk. The first one is, what is the emission rate for the various sources and activities? Because it's very important that we realize the exposure to bioaerosols will occur without us noticing it. It's very transparent, it's invisible. If you are questioned on what are you eating, what water source are you using for consuming, it's easy to find an answer to that. But if I'm asking you, when you're outside at your home, where does the air you breathe come from? It's very puzzling. We have no clue what we are breathing. So it's very difficult to, and if we don't have any data for emission rates, it's very difficult to pinpoint the major sources around you. The dispersion studies are also missing. So how does a source emit and how do the bioaerosols disperse in the environment? And how can we mitigate that? Because with the air, what's very interesting is we can eventually think about a way to prevent emission outdoors and prevent the spreading of those genes in the environment. So in 2019, NSERC launched is a research Discovery Frontiers program. So if you're not familiar with that program, I don't think there has been another launch since 2019. But in this competition, they funded one team in Canada, and the overall topic was AMR in the environment, and we were fortunate enough to be funded by that program. So it's a 4-year program, and this is the overview of the program that we have proposed. So we proposed to look at the various sources of ARGs in bioaerosols. So we looked at agricultural buildings, fish farming, manure spreading hospitals, wastewater treatment plants. Run some dispersion modeling, look at some passive sampling strategies, also look at very remote places where we could find those ARGs in the air, so far north of Canada, and also we looked at the clouds. We also did a transatlantic survey. We used a car filter survey to do a trans-Canadian mapping of the ARGs. Of course, estimate the exposure of people through the aerosol route. Run risk assessment models, run animal models, and also run culturomics to evaluate the whole genome of bacteria found in agricultural environments. So this is the big picture of our project, and we published a paper just presenting the idea of the project, because the reason why I'm very happy to be here this morning, it's because we want this project to be known, we want you to become interested, but we also gathered thousands of samples in the freezers that we are very happy to share with any person who would like to move forward with those samples and eventually run more analysis. So unlike the other types of matrices that we sample, water, soil, human, sampling the air is very challenging because it's very diluted and we have to concentrate. So we managed to find a standardized sampling protocol that would be used throughout our project. This included very high volume air samplers, such as this one that samples 4 cubic meters per minute. So it allowed us to retrieve highly concentrated samples from even outdoors. So the philosophy behind the project was also to develop a standardized protocol for the sample analysis. So we developed a protocol for qPCR for ARGs and mobile genetic elements that would be performed on all our samples throughout the project. Of course, run a 16S amplicon-based for biodiversity and network analysis. Characterize the ARG structure and context using culture, so performing whole genome sequencing on isolates from some of those sources, and also calculate concentrations and emission rates when it's possible. So when it's possible to use the source as a chimney, so as a point source of emission of ARGs, we would try and calculate emission rates, then we could evaluate the modeling dispersion. So we used this platform. We ended up, as a teamwork, we decided that we would target 37 ARGs, 3 mobile genetic elements, reference genes also. So this is exactly what was done in all our samples. But of course, as I said, we still have all those samples in the bank, so we could run other analysis. So the first part of the study was to characterize the sources and developing also sampling methods. So we looked at agricultural buildings with and without antibiotic, but also we try and develop a sampling approach at the fan exhaust instead of going in the barn to see, you know, with the outbreaks, the viral outbreaks in swine barns, poultry barns, it's very difficult sometimes to enter the premises. So we have developed an efficient air sampling approach at the fan exhaust that could give a qualitative idea of what is being emitted from the source. So the impact of antibiotic use is also analyzed. We studied fish farming, but for now we don't have, I will really focus today on the parts that are published. Manure spreading activities, wastewater, and hospitals were part of the targeted sources. So Joanie, she'll be presenting a talk, it's here. I don't know where it is. It's here, yeah, May 28th, she'll be presenting. But just as an overview, she managed to determine, like, how can we sample at fan exhaust, outdoors at different points, like on the map here, to downwind, upwind, to evaluate, like, the gradient of concentration, and eventually run dispersion models with those data. She also ran analysis using culture in antibiotic-using and not antibiotic-using poultry and swine farms. And just to give you an idea, she could calculate the emission rate of total bacteria from those barns to up to 10 to the 11 per hour. So this is just an example of how much, like, stuff those buildings are emitting. And this is possible only because we're collaborating with engineers, with the people at IRDA, and they characterize the building, the ventilation rates, and they do all that stuff so we can eventually transform our concentration data into emission rate data. One other interesting thing that we did is we looked at the passive sampling strategies. So what if people out there are interested in looking at what ARGs are floating around but don't have access to air samplers, to sampling strategies, very complex and costly? So Samantha here has used conifer needles as passive samplers around farms, but also other types of passive samplers. And she has already published one manuscript, but she's also presenting during this conference a poster on her recent findings. But it's very interesting that those pine needles, like, are memories of what has been floating around, like, for a long period. So it's very interesting. We could gather a lot of data with very cheap sampling strategies, retrieving pine needles or conifer needles. And it's so far, I think, a very promising approach. She also did that close to barns, so in the trees surrounding the barns, and compare it with volumetric sampling. So we can really compare, like, real air sampling as well as pine needle samplings and passive air sampling. So this is very promising. One other activity that we were very interested in is manure spreading, and we all smell it. I don't know if some of you live close to agricultural fields, but when we spread manure, we can smell it. But what if that smell also contains bioaerosols? But it's very challenging to sample during manure spreading events because it's, like, long-term on big fields. It disperses in all the dimensions. So we did that. We also looked at the trees surrounding. This tree line was planted for our project. So during, like, experimental manure spreading, we could compare, like, active air sampling, the needles as well. But we managed also to use, like, a lab, an experimental manure spreading, like, setup. Natalie will be talking to you about the challenges of sampling outdoors during that project, and I think it was a huge challenge because, of course, aerosols are not, like, running towards our air samplers. We have to be very strategic. So we had access to this, and I will discuss that in more details during the workshop on Wednesday, the workshop 7. So this is a big, like, a greenhouse that was built and designed by our engineer friends. And we could spread a known amount of manure in a certain amount of minute and then sample at the exhaust. This is a swine, and the previous one was probably cow manure. So we could sample at the exhaust here, and all the air from the building is exhausted by those fans. So the good aspect of that setup is that we have access to all the air that is circulating horizontally in the building. So actually, we have access to all the bioaerosols emitted during the manure spreading. So everything is enclosed. Besides doing something like this, it's impossible to evaluate the emission rate of sampling. We did some calculations, and we ended up with, like, it's very probably an underestimation. So the province of Quebec, here I'm saying that we spread 1 billion kilograms of swine manure every year, the same amount of cow, the same amount of poultry manure. And it's not 1 billion now, I think it's 10 billion. I looked at the most recent numbers, so it's underestimated. And each kilogram of manure emits 1 billion of ARGs in the air. So if you calculate, if you do the maths, it's possible that only for the swine manure in the province of Quebec, 10 to the 18, 10 to the 19, or 10 to the 20 ARGs are emitted in the air. And as I said, we smell when we spread, so it's very likely that the surrounding areas also are exposed to huge amounts of antibiotic-resistant genes during manure spreading activities. So for wastewater treatment, it was more challenging, and thanks to a collaboration with Émilie Bédard at Polytechnique and her team, we managed to sample inside an air treatment unit in a wastewater. So this paper really is a proof of concept that we can sample within a ventilation duct, kind of. This time, this is an air treatment system. But we can have access to whatever would be emitted outside if there was not such an air treatment like at the fan exhaust of those wastewater treatment plants. So it's very interesting. We can evaluate the efficiency, so the mitigation efficiency of those air treatments, but also evaluate the emission, the potential emission of wastewater. So now we know there's stuff emitted in the air, so where does it go after that? So we were very interested in long-distance transport of those ARGs emitted from point sources. So the different topics that were addressed were Canada mapping using core filter, Canada North, clouds, transatlantic, and dispersion modeling, and I will focus on a few of those areas. So my colleague here, Paul Georges, was in my team then. Now he's flying like an adult bird. He's now a professor, so I'm very happy. So Paul did a postdoc in my lab during COVID, and he managed to retrieve, there was a similar study that was performed by our Chinese colleague Mao Shengyao a few years ago. There was no Canadian city involved, but now he managed to retrieve almost 500 car filters throughout Canada, and also develop an extraction protocol from those air filters, you know, and air filters from cars, it's garbage. People throw it away. So we could retrieve it like in large amounts and have a very, very detailed mapping of the country if we could like just have some garages involved, and it's really community science that could be happening here. So he's extracting the DNA from that, good quality material, and he really managed to include some population metrics, hospital data, wastewater, etc. So he could really correlate the position of the air filter, where it comes from, and what it contains as far as antibiotic-resistant genes. So Paul will be also presenting a poster about that this week. For the clouds, we're collaborating with a French team, and Florent Rossi is a postdoc in our lab. So we've been sampling clouds, sampling also aerosols in this summit here. So this is a very high altitude summit. It's a research station. So we also did the overseas samples that I won't be talking about, but antibiotic resistant genes were analyzed in those samples. So for the cloud study, Flora managed to sample 12 clouds. So we know exactly where the clouds are coming from because the back trajectories are available. So it could compare, like when it comes from the continent and when it comes from the ocean, how is the ARG profile different? So it's very, very fascinating to realize that per year, up to 10 to the 25 ARGs are circulating through the clouds. So then we can imagine that there is no boundaries, no no differences between countries. So if the neighbor country uses a certain type of antibiotic in agriculture and the ARGs are circulating, it can come up in the clouds and fall back as rain later in another territory. So it's very global. Their impact is very global, and understanding that is very interesting, I think. But also now we have the data from 2020, 21. Now we can follow up during the next years and see how this evolves and how it is changing according to the new practices that are being put together in various places on the planet. The dispersion modeling is going on right now. I don't have any data to show you, but what we want to do is really assess how far can ARG travel from a point source, and how can it impact the surrounding homes and the surrounding people, and how can we eventually give this information to city people to see how we could design like the places and how we could minimize human exposure using those dispersion models. One aspect of our project that is very interesting and it's unique, it was to start with EdTop was part of our Frontiers team to start with. He's still involved through the management committees of some students. And he had this wonderful idea of using animal models to study how can we study the transfer of ARGs from an air sample to the gut of a mouse using a model. And we're fortunate enough to have André Marette, who is a colleague in our research center, who is an expert in obesity and microbiota. And his student, Ilaria, will be presenting also today. And she's really looking at working with Joanie, taking air samples from barns and concentrating the bacteria and then feeding the mice with different food and different bacteria and see how like the transmission can occur in the gut of the mice. So this is also very interesting. Also, we have some collaborators that are experts in risk assessment models. Jan Parmley at Guelph is responsible for that aspect of the project, and her student Manuel is also presenting here during that conference. So this is this team that will take over our data and try and implement it in their risk assessment model to see how can we interpret the exposure data to bioaerosols containing ARGs into the risk assessment models actually available. Now we know that some activities emit bioaerosols, so what do we do with this? So one very important aspect of our project is mitigation. So we just don't say, oh, it's very sad, we're emitting ARGs everywhere, we have nothing to do with it, and we cry and we are completely depressed. No. We're trying some stuff. So we are fortunate to work with engineers, as I said before, and some people that have very broad areas of expertise. And our agricultural engineers have tested like different types of manure spreaders. So if we inject the manure in the land instead of spraying it on top of the land, how can we impact what is emitted? Can we bring it a little bit lower? Can we reduce the emission from the activity? How could we use trees around some buildings to mitigate also the spreading? This is something that will be studied during Samantha's PhD studies. Air filtration in hospital. We have access to air filters from filtration unit in the hospitals. What is in there and how can we improve the filtration efficiency and how can we reduce what is emitted? So as I said before, we have a huge bank of samples. Everything was entered in a database. So we have for each sample the site, the geolocation, the sample nature, etc. So we think that it's a huge opportunity for collaborating with whoever is interested. So it's in RedCap, it's like this, but if you're interested, we could certainly send you an invite so you could have access to the database. So we know that ARGs are widely dispersed from several human and animal activities and may contribute to the risk of developing AMR infection. We have characterized ARGs in various bioaerosols with significant emission potential, and everything is banked, so it's available for collaboration. Sampling outdoor is very challenging, and passive air samplers are very promising. The fan exhaust can be used when entering the facilities and is not an option. A wind tunnel has a high potential for emission studies. I showed you for manure spreading, we could do whatever activity in there and calculate the emission rates. Dispersion model will lead to exposure potential in high-risk zone. So we could certainly, after that, focus on the eventual risk for disease in humans. So I wanted just to present the people that collaborate to that big initiative. Of course, I don't have the time to go through that. The professional, the research professional, and the experts, and the highly qualified personnel, so the students involved also, and the beautiful team from my lab since the COVID to now. So my website is there if you're interested. Thank you so much for your attention.
And that gives me the pleasure to introduce the next speaker. And the next three speakers will have not so much time to present, but the next speaker is Marko Virta from the University of Helsinki, who will present on antimicrobial resistance dissemination potential of bio-based fertilizers according to field trials. Go ahead, Marko.
Good morning and thank you very much, but I will present this data that was produced by Johanna Muurinen, who was in fact supposed to also present it, but unfortunately she couldn't participate, so I will try to manage. Mineral fertilizers are very much part of the world's pollution crisis, and in fact nitrogen and phosphorus, they may be one of the biggest problems in our pollution crisis. And these, when we think about mineral fertilizers, these macronutrients, they are combined with micronutrients to produce something like chemically defined fertilizers. And for those, phosphorus comes from rock, so it's a limited resource, as we know. Nitrogen comes from air with a very energy-intensive process, so very much natural gas is used, which results to high carbon emissions. Also, when we think about the global nitrogen and phosphorus suppliers, we can easily see that in top 10 there are no countries in Europe, so Europe should think something else. In fact, there is only one phosphorus mine in EU, that's in Finland. So the question is how to increase sustainability of these nutrients and also how to kind of make EU more self-sufficient with these nutrients. One possibility is to use bio-based fertilizers which would be made from nutrient-rich side streams. And for that we had Lex for Bio, EU Horizon 2020 project, where we first studied what side streams are available for possible bio-based fertilizers. And after finding those on EU level, we in the project studied the effect of the selected fertilizers for soil quality, then agronomic efficiency, how the BBFs work as a fertilizers, and then risk assessment for heavy metals, organic contaminants, and effect on AMR. These were studied in the field experiments in 3 different countries. In Finland, here quite north of Europe, close to Helsinki. Then in France, near Saint-Malo, which is here northwest of France, and then in Spain, in Seville. So we covered different climatic and different soil conditions with the fieldwork. In the project, also different socio-economic issues were analyzed. Here I'm presenting only the AMR results. But before going to results, we can first have a look at these bio-based fertilizers. So I already told the idea behind the mineral fertilizers. And when we have a look at these bio-based fertilizers, we can quickly figure out that the system is very different. So there are some side streams that can be used for making fertilizers, and that's what we get. In the project, we divided the fertilizers for two groups, for phosphorus fertilizers and nitrogen fertilizers. But there is clearly overlap in these, and this was more like a practical solution. We made a selection for 15 bio-based fertilizers that were tested in each country. In addition, for each country we selected local fertilizers that are not available in big quantities but may be relevant for that country. And we also compared the results with mineral fertilizers with two different application levels. There were separate field experiments for phosphorus and nitrogen fertilizers in these three countries, and there were always 4 plots per fertilizer in each field, and those form kind of a biological replicates. Then we sampled soil before the application of the fertilizer, immediately after the application, and 6 weeks after the application, and made Illumina metagenomes from those soil samples. Here are a couple of figures from Finland. So this is when the fertilizers is applied, and this is after 6 weeks. And this is Johanna processing the soil samples in the lab. So this was during the peak of pandemic, so we were not able to visit in different countries, which was a little bit of a drawback. We studied soil with metagenomes, and we also studied the bio-based fertilizers themselves with the qPCR array run by Resistomap, just to find out what if the fertilizers themselves contain resistance genes. But the main idea was to analyze effect of the fertilizers for the soils. So we did it by Illumina metagenomes, and we analyzed microbiome, resistome, and mobilome. Then to the results. First, the resistance genes in bio-based fertilizers by qPCR. So it's easy to see that the fertilizers, they form 2 separate groups. The group in left had very low abundance on almost all tested genes. On the other hand, there is another block of fertilizers where there is a quite high abundance for many different resistance genes. And this group is even divided to 2 kind of subgroups. The ones presented on red are based on poultry waste, and they contained many gut-related antibiotic resistance genes and mobile elements. They may also contain antibiotic residues based on the results from the other work buckets of the project. So they may cause, in fact, quite similar risk than using poultry manure as such causes. Another group that had high abundance of different resistance genes is this blue one, which are composts and digestates, and for a little bit surprisingly, they contain resistance genes against, for example, carbapenems. So it's kind of a interesting story, but I cannot tell more about that, at least not yet. Then we can go to the soil results. And this is quite a busy slide, but I will try to first go through how to read it. Here, in the first column, we have always the result metagenome from soil before application of the fertilizer. Then this represents the result of the application, before application of the fertilizer. And then there are 2 samples. On left side there are metagenome results right after spreading the fertilizer, and then on the right side 6 weeks after. And they are all in the same way in all panels. And in top there are results for phosphorus fertilizer, so that on left there is a diversity of antibiotic resistance genes, and on right there is some abundance of antibiotic resistance genes. And in below the same for the nitrogen fertilizers. So especially if we look at the 6 week after results, we can see that there is very little effect after 6 weeks, maybe with one exception, which I will come back later. So this was the results from field experiment in Finland. Then the same for France. Again, there seemed to be very little effect after 6 weeks, with one exception. Then the Spain. Here also the influence was small, but there may be some exceptions here, maybe more than one. But I must stress here that the overall effect on the soil was low. Then we can go through these exceptions. So first is 6 weeks, which is maybe the most drastic one, because it's somehow lasting effect. Maybe it's a bit surprising that the one that has highest some abundance is in fact plant-based and after fermentation. We don't know why yet. Then these other two, they are poultry-based, and then again there are some more poultry manure-based, bio-based fertilizer showing up in the figure. Then if we think about right after the application of the manure, we have again some poultry manure. Then something that is just said that it's animal byproducts. Some manufacturers don't give very much details. And then there's a pig manure. Then to conclude, I think the main conclusion is clearly that bio-based fertilizer in general, they have low AMR dissemination potential, with poultry production-based BBF maybe some exception. Also, I was not showing, but we learned that the bio-based fertilizer have no lasting effect on soil microbiome. And then we think that this is valuable data for understanding the ecology of AMR in the agricultural soils, because we had quite large field trials in different climate and soil conditions with quite large number of metagenomes, and we also have data from different methods, from different work packages of the project available. So next step will be assembly of genomes from the metagenomes, especially on the high ARG samples, and then analysis of genes encoding metabolic pathways, especially for carbon and nitrogen, and combining the results with others one. So my time is up, so thank you.
Now it's my pleasure to introduce Jan Siemens from the University of Gießen in Germany, who will be talking about replacing untreated and with treated wastewater for irrigation, consequences for antibiotics, metals, and nutrients in soil. Lovely to meet you.
Thank you. So why is this interesting for us? Well, the growth, especially in large urban areas and mega cities, the rising food requirement of a growing population, but also increasing water scarcity aggravated by global change, increase the pressure to use wastewater as a resource. And in our preceding talk, we had the reuse of nutrients that can also be achieved with wastewater irrigation. So there's an increasing pressure to use wastewater for irrigation purposes in agriculture, and this is done over many places of the world, and also often polluted river water containing wastewater is used for irrigation of agricultural land. One of the most prominent examples of use of wastewater for irrigation purposes is the Valle Mezquital north of Mexico City, where some 60 cubic meters per second of wastewater are used for irrigation of more than 900 square kilometers of agricultural land. And together with the wastewater come nutrients and organic matter that is contained in the raw wastewater. And this increases the fertility of the soils, as you can see here, the green color, the vigorous plant growth there. And by increasing the plant growth, also the input of roots and plant residues in the soils are increased, which leads then to a doubling of the soil organic matter content in these soils over time, which is good because this soil organic matter or humus helps to bind and detoxify the pollutants that also come into the fields with the wastewater, like heavy metals, or in this case, sulfur methoxazole, which in concentration increases over the time of irrigation as well. So to reduce health risks associated with wastewater, large investments have been done and are underway to reach Sustainable Development Goal 6, clean water and sanitation. And in the Valle Mezquital, the Atotonilco wastewater treatment plant has started operation in 2018. It is the third largest wastewater treatment plant in the world. It has cost more than $800 million US. It's the largest one that has ever been built in one go. And the main task of this wastewater treatment plant is, of course, to reduce the number of bacteria in the wastewater that is used for irrigation, but it also reduces the load of organic matter and suspended solids in the wastewater used for irrigation, as can be seen here in this picture of bottles on the car of Christina Siebe. Here you have the untreated wastewater, agua negra, as the Mexicans term it, and this is the treated wastewater, so much less organic matter and suspended solids. But what consequences has now the implementation of wastewater treatment for the wastewater irrigation system? So we have learned that in the past 100 years, irrigation with untreated wastewater has increased the soil organic matter levels, but also the concentrations of pollutants in the soil. So when we now start using treated wastewater with lower contents of organic matter and nutrients, we fear that this will decrease soil fertility, decrease organic matter inputs, which might lead to decreasing soil organic matter contents, which might open a window of elevated risk in this transition period in which pollutants that have been accumulated in soil are released. So our hypotheses were that replacing untreated wastewater with treated wastewater causes a release of pollutants that have been accumulated in soils in the past, and that irrigation with treated wastewater reduces the availability of nutrients in soils. And these hypotheses were tested in an incubation experiment. So we collected 3 different soil types: leptosol, shallow, sandy, stony soils, phaeocims, and vertisols, so very deep and clayey soils, 4 fields of each soil types in the Valle Mezquital. And this soil was shipped to Germany by plane, together with treated and untreated wastewater in cooled transport. And then in the lab, part of this wastewater was also spiked with antibiotics and disinfectants to increase their concentrations. And afterwards, this wastewater was mixed with soil in sterilized honey jars. And these honey jars were then covered with face masks, of which we had a lot during the COVID pandemic, and incubated at 22 degrees Celsius in climate chambers, and then sampled at after different periods of time of incubation. When we go to the concentrations of antibiotics and we look here for sulfur methoxazole on the y-axis and time here on the x-axis, we don't see a big difference between concentrations of sulfur methoxazole that we can extract with water, so that are water soluble and therefore accessible or effective for the soil bacteria. As you can see here, the blue dots representing the soil mixed with treated wastewater is very similar to the greenish brown dots representing the soil that has been mixed with untreated wastewater. This picture changes when we now go to the variant in which we have added antibiotics and disinfectants. So when we have a look at the soil that has been mixed with untreated wastewater first, we see that, so where's my red dot? We see that directly adding the treated wastewater that has been spiked with sulfur methoxazole, we cannot extract this with water. So perhaps the sulfur methoxazole we added was bound to the particles contained in the untreated wastewater and to the organic matter, so that it didn't show up in the water extract. But then quickly after 6 hours, we could extract it with water because it was released from these particles. But then very quickly a dissipation of this sulfur methoxazole started with a half-life of 3 days. The picture was different when we added the sulfur methoxazole to the treated wastewater. So we could extract it immediately with water from the soil, and then during the first 2 days, the concentration even slightly increased. And then after this lag period of 2 days, a slow dissipation started with the half-life more than almost 3 times as slow as the dissipation with untreated wastewater. With regard to the metals, we had the interesting observation that for the lactosols and the phaeocyms, there was not much big difference, but for the clay vertisols, after 14 days, the water-soluble concentrations of copper were higher in the soils that were mixed with treated wastewater than in the soils that were mixed with untreated wastewater. And my hypothesis to explain this pattern is that it has something to do with the availability of nutrients and organic carbon. Because copper is very strict and very strongly bound by organic matter in soils. And when we now move to the availability of the nutrients of nitrogen, we see that here in these clay vertisols, the picture is just the inverse of the picture that we saw for copper. So my theory is, that has to be tested in further experiments, that the microbes here, the soil microbes, after 14 days, they ran out of nitrogen because we added less nitrogen with the treated wastewater, and they started mineralizing and mining the organic matter for nitrogen, thereby releasing the copper that was bound to that organic matter. So what we have learned from this incubation study is that indeed treated wastewater reduced nitrogen and increased copper availability, not in all soils, but in the clay vertisols. That we didn't see a mobilization of antibiotics, but a slower dissipation of sulfur methoxazole added with treated wastewater, again in the clay vertisols. Overall, soil buffered the effect of the wastewater treatment on pollutants, bacteria, ARGs, and MGEs, as can be also seen in the posters of Laila Soufi and Sarah Gallego that will be presented by Connie Smalla in the poster sessions, and Katharina Axtmann. So, which leaves us with the question of that whether irrigation with fresh produce with treated wastewater is really safe. Well, and with this, I want to thank you for your attention, the German Research Foundation and UNAM for funding, and invite you also to join us because we have an open postdoc position.
Now it's my pleasure to introduce Thomas Behrendonck, who comes from Technische University in Dresden. and he will be giving a talk on trade-offs of increasing temperatures for the spread of antimicrobial resistance in river biofilms.
Thank you very
Thank
much.
you very much.
Originally, this talk was supposed to be given by our PhD student, Kenyum Bagra. She's back in India. Unfortunately, she didn't get a visum. So it's mostly unfortunate for you because you cannot listen now to a young, dynamic, really very talented PhD student. Instead of that, you have to listen to an old white man. So what we present is on the trade-offs increasing temperatures for the spread of antimicrobial resistance in biofilms, and why did we do that? So the motivation was, we know that increasing temperatures are happening. We are all aware of climate warming, and we know its effect. We also know that communities change due to climate warming. And we also know that many pathogens have their temperature optimum rather at higher temperatures. So for us, it was the question, should we ask some simple hypotheses to understand what this means for the dynamics of antibiotic resistance in the natural environment? And therefore, we have formulated three rather simple hypotheses. One is if bacterial community and resistance change due to temperatures may increase, and if this changed bacterial community then is less resilient to invading resistant bacteria. In our case, invading bacteria are usually wastewater treatment plant effluent bacteria. And we also then assumed, or that was our hypothesis, since the invaders like E. coli have a higher growth optimum at higher temperatures, should increase. To study this, we have developed an experimental system which we like a lot. And when I say we, this is actually the Anti-Resist Consortium. Many of them are present here. And I can only recommend to go also to poster 14 of Christoph Möller and listen to Uli Klümper's talk where you see the system as well. So what is that? Essentially, we expose glass slides into the river so that a natural biofilm can grow on it. And you see that here we have several ones so that we can work in a replicated manner. And it's heavy enough so it doesn't get removed. And after we have exposed it, we take it into the lab, and then we can consecutively remove the slides with a biofilm and analyze that. It's a recirculating system, that's important to know, and it's recirculated with the water of where we have removed the slides. Now, we have then used the system and exposed it to 3 different temperatures, 20, 25 degrees, 30 degrees. So simple, no fluctuating temperature or anything like that, just constant temperatures. And then we analyzed the biofilm 1 week after it has acclimatized to the temperature. And then again, 1 day after, we gave it 1 pulse of 20% wastewater, second day, 7 day, and then also 14 days later. And some of the slides I only show you the first and 14 days. Initially, we did high throughput qPCR to analyze it with resistomap and then chose selected numbers of resistance genes, as you will see in a second. And we also used 16S to characterize the bacterial community, all standard analyses. Now what you see here is 3 slides. One is an NMDS plot, and you see the different temperatures indicated by the different colors again, and the numbers mean the days when we sampled. And so what you see is that the community changed over time, but it stayed distinct according to the different temperatures. And that is essentially what you also see in the B slide. What you see in the C slide is the abundance. So it's just the number of 16S copies which we used as an indication for bacterial abundance. And you see that there was not a big growth of bacteria or biofilm growth. It rather stayed the same, independent of the temperature. This was our first surprise. Our next surprise was that when we now just looked at the biofilm, there's no wastewater exposure, nothing. It's just incubated for 1 week at different temperatures. And there we quantified the selected resistance genes where we saw differences according to the high throughput one. And you see the relative abundance for different resistance genes, and the different colors indicate the different temperatures. Here are the colors and the temperatures. And on the top right, you see the wastewater. Yeah, so we see many more or higher concentrations of resistance genes in the wastewater. But what we also saw is that after 1 week in the 30-degree treatment, we had a significantly higher number of resistance genes. And this is just of the biofilm which we just took from the river, nothing else. Then this is the analysis now after we had pulsed it with wastewater, 1 day later. And here you see, here you see in blue the control and in red the exposed ones, and on top the different resistance genes and some taxonomic markers as well. And most importantly, here are the 3 different temperatures. And what you see is that you don't see really much, except that in all exposed units we get a higher concentration of resistance genes, independent of temperature. So that was also unexpected for us. And then when we looked at it after 14 days, this is the same slide now again, the different temperatures again. And now if you look at the lower panel, you see that there are no significant differences anymore. So at 30 degrees, it seems that the community went back to the original assembly, if you want. But at 20 and 25 degrees, we still could observe a higher concentration of resistance genes and also of the focus bacteria, which we were looking at, E. coli and enterococci. So this was not at all what we expected. If we look now, you see that even though we had higher numbers, it didn't mean that they all grew. It was just a difference in not growing, if you want. You have to keep in mind, we pulsed it once with a treatment plant, and then there was no nutrient available, just what's in the river water. So I think we believe it's a nutrient-limited system. And what you also see here, and this is now the panel, it's a bit different. Now you see the different temperatures up here, and here are the different resistance genes, and here's the time over the different days when we sampled. And we saw that the decrease of the resistance genes was lower, was higher at the 30 degree, and for 20 degree we had a lower one, and even stabilizing, for example, for some of the resistance genes at the lower temperatures. This is also nothing what we really expected. We expected this effect to be much stronger at 30 degrees. And if you look again at our candidate bacteria, we saw a similar effect. There we also saw that the decrease was different between the 30 degrees and the 20 and 25 degrees temperature. And if we now look at the microbial community again at day 14, we again have the same NMDS plot as I showed earlier. But now you see, for example, the community of the wastewater is on the top right here, so it's, it's stayed different. And if we now compare again the 20, 25, and the 30 degrees, you see that at 20 and 25 degrees, the community stayed distinctly different. Yeah? So the colors don't overlap so much, blue and red. Yeah? And 1 and 14 are again the days. But at 30 degrees, we notice here that the communities became more similar again. So what does that mean for our hypotheses? That means that the bacterial community and the resistance genes do change to temperature increase, but we only observed this at 30 degrees initially, when there was no wastewater present, if we remember. And what we also observed is that the change, due to temperature changed bacterial community is less resilient to the invading bacteria, except for 30 degrees. There, the community was quite resilient. And we believe that this is due to the fact that the biofilm could grow faster at the 30 degrees, and therefore all niches were occupied at a higher abundance, and therefore the invading bacteria could not be, or resistance genes could not be as successful. And then our last hypothesis, that the invaders with a growth optimum at higher temperatures will increase. No, they decrease at all temperatures, but they decrease less at the colder temperatures. And that was also not necessarily something what we expected. Of course, these are quite simple experiments, but I think they give a good indication that you might expect results which you don't expect. We will get results you might not expect, due to the fact that the community is actually, so the resilient community is determining the success of the invader or the resistance gene. Yeah, with that, I'm in time and at the end. And again, thank you to Kenyum. We are really sorry that she cannot participate. And of course, also thank you to my colleague, Gagi Singh, who is the co-advisor of Kenyum, and of course, David and Uli, they are both here. So if you have further questions, you can also ask them. You can, of course, also ask me. And Resistomap and all the funding agencies who helped to make this possible. Thank you very much.